This lecture talks about the process of meiosis, which will decrease the chromosome number by half. So terms that you need to know for this unit for meiosis include haploid, which means that the cell has one set of chromosome. So these would be sperm and egg cells, the gametes. Diploid, which is represented by 2N, has two sets of chromosomes, and these include somatic cells. So in humans, all our somatic cells, all the cells of the body besides sperm and egg, have 46 chromosomes, but the haploid cells, the sperm and egg, would have 23. Polyploid means that there are more than two sets of chromosomes. So for example, some plants are polyploid, have multiple sets of chromosomes. A sister chromatid is an identical chromosome that is attached to its identical chromosome at the centromere. So when one chromosome replicates itself and it's identical, then it will have a sister chromatid. And then we talk about homologous chromosomes, which is actually a pair of chromosomes that carry genes for the same traits. We have two chromosomes that have the same genes for all the traits. And a tetrad is a structure that consists of two homologous chromosomes that are bound together, or four sister chromatids. So tetra means four, so four sister chromatids. Now if we look at the steps of meiosis, we've learned about mitosis before, and the first phase is interphase 1. Meiosis is going to be divided into two phases. So interphase 1 is when DNA replication happens. So the chromosomes are duplicated, cell size increases, the organelles are replicated. And then prophase 1, each chromosome is going to pair with its corresponding homologous chromosome to form a tetrad. This is different than mitosis. So when this happens, we're going to talk about a process called crossing over, which is going to increase genetic variation. That may occur at this time. So now we have this tetrad, and the nuclear envelope disappears, centrioles are going to move to opposite sides of the cell, and then just like in mitosis, we go into metaphase 1. The spindle fibers are going to attach to the chromosomes, and the chromosomes are going to line up in the middle of the cell. Anaphase 1 is when the spindle fibers are going to pull the homologous chromosomes apart towards opposite ends of the cell. And then in telophase 1 and cytokinesis, the nuclear membrane forms and the cell separates into two haploid cells. In meiosis 2, we skip interphase now because we have those two cells, and each haploid cell, which has the same number as the original cell, undergoes meiosis 2 without DNA replication. We have all the DNA that we need. The nuclear envelope is going to disappear, and centrioles move to opposite sides of the cell, just like they did in prophase 1. In metaphase 2, the spindle fibers again attach to the chromosomes at the centromeres, and the chromosomes are going to line up in the middle of the cell. In anaphase 2, the spindle fibers then pull the sister chromatids apart towards opposite ends of the cell. And in telophase 2 in cytokinesis, we have the nuclear membrane forms again, and the cell separates into four cells. Now, we're going to get four equal sperm cells, or one egg cell and three polar bodies. But each number is going to have half, okay, true haploid, little n, number of chromosomes. If we look at gamete formation, what this process is used for, here's spermatogenesis. We start with these four chromosomes here, and what happens is they replicate during interphase 1, and we get the homologous, we get these sister chromatids. You see the X's that we're used to, and we're going to get a tetrad here. They will eventually separate, so you have each of those chromosomes attached to its sister chromatid in the cells, and they divide again in meiosis 2 now to form four haploid cells. So we have four cells with half the number of chromosomes. And notice that in spermatogenesis, we end up with four equal-sized sperm cells. In oogenesis, gametogenesis in females, we start out the same way, but what happens is the egg cell is much bigger than the sperm cell, and it happens in this process. There's going to be one cell that's actually going to have the majority of the cytoplasm, and that's going to be needed in order for that embryo to develop. So the sperm, in comparison to the egg, is quite small, but it still has the same number of chromosomes. So in oogenesis, we're going to get one egg cell and three polar bodies, which are these smaller cells that are going to be discarded. These aren't going to be used for reproduction. If you remember in the video when we watched Life's Greatest Miracle, that it talks about, there was a quote that it's quality, not quantity. And this is what they're talking about. So one mature egg cell is going to develop, which will be fertilized or has potential to be fertilized by an egg, whereas these three polar bodies are much smaller and don't have enough to support the process. And then if we compare meiosis and mitosis, here in meiosis, we end up with homologs or homologous chromosome pairs. They're going to meet up and form tetrads. You can see we have four legs here. Um, and those tetrads are going to be split in one division. And then the second division, meiosis 2, after we have the two daughter cells, then it's going to look like mitosis, and we'll end up with four haploid cells. And these are not going to be genetically identical to the parent because of crossing over and another process-independent assortment, which we'll talk about later. In mitosis, we have our chromosomes, they line up in the center, and the sister chromatids are pulled apart. This would look like it would in meiosis 2. And then we have two daughter cells form, and those are genetically identical to the parent cell. So that's the big difference between meiosis and mitosis. Mitosis, we get two identical daughter cells, and then in meiosis, we get four non-identical cells.